Welcome back, everyone, to the Connect Science webinar. It's a distinct pleasure to welcome Scott Armstrong. Scott is a professor of pediatrics at Harvard Medical School and the chair of the Department of Pediatric Oncology at Dana Farber and Boston Children's Hospital. As everyone knows, Scott has made seminal discoveries related to the role of MLL rearrangements in acute leukemias, but also other aspects of epigenetic alterations across multiple different types of cancer. Just housekeeping announcements. You're all welcome to post questions using the Q&A tab on your screen. Please don't post them using the chat tab. We'll probably just ask you to repost them using the Q&A. That way we can keep them together. Time allowing, we'll address as many as possible at the end, and Scott will have the opportunity to review them and respond to ones we don't have time for. As always, we'll be skipping Wednesdays because of an internal Dana Farber meeting. On Thursday, we're lucky to have Marcus Munchen from the City of Hope presenting on his work related to metabolic deregulation and B-cell malignancies. So I don't want to delay any further. Scott, please take it away.
Well, thank you, David, and thanks to you and to Nellie for hosting this series. It's really been a terrific way to keep the scientific community together during this time. So these are my disclosures. I am consulting with a number of biotech and pharmaceutical companies around small molecules that target chromatin complexes, but none of which are relevant to the talk that I'm going to give today. So broadly, the talk is going to be focused on two main questions that are highlighted here. And on the left, you see the hematopoietic hierarchy ranging all the way from the long-term hematopoietic stem cell, which most will know that is the only cell type that has long-term repopulating and self-renewal potential in the hematopoietic system, to the multiple different cell types that can arise from those hematopoietic stem cells. And one of the questions in the lab, and that many of us have studied, is which of these cell types are potential cells of origin in leukemia, and how do gene expression programs during the transformation of given cell types to leukemia change? What are the critical changes in gene expression, and are there differences in the types of leukemia that arise from different cells of origin? And of course, these same concepts are relevant for a number of different types of cancer. So I'll talk a little bit about that. And on the right is the MLL protein complex, which regulates chromatin. And we will talk a fair amount about some of the details around chromatin-associated complexes and how we're thinking about targeting those complexes, again, mostly in leukemia, but at the end, I'll talk a little bit about potential opportunities in other diseases as well. So as David mentioned, my lab has been interested in how it is that translocations involving the MLL gene on chromosome 11 Q23 lead to the development of leukemia. And here is shown the first published karyotype of a ALL case with a 411 translocation. You can see here is chromosome 4, and much of chromosome 4 has been lost and translocated to chromosome 11. So this was just one case in 1977, but this was during a time when Janet Rowley, among others, Nolan Hungerford, were really showing the importance of BCR-ABL rearrangements, and the MLL rearrangements were found soon thereafter. Then throughout the 80s, a number of studies showed that the MLL translocation, or in this case, the 411 translocation, was recurrent in ALL. In the early 80s, Clara Bloomfield and Janet Rowley demonstrated that these translocations tend to be associated with infants with leukemia. We now know that, in fact, adults with different subtypes of leukemia can have 411 or other 11q23 rearrangements as well. And then in the mid-80s, a number of pediatric oncology groups demonstrated that the presence of this translocation, that is rearrangements of chromosome 11 in position Q23, predict a poor prognosis, particularly in infants with leukemia. And during that time, it was also found that both pediatric cases and adult cases of acute myelogenous leukemia also could have rearrangements of the 11q23 gene. And then in the early 90s, during a period when chromosomal translocations, the cloning of the breakpoints of chromosomal translocations was really starting to happen, or a number of groups shown here, 4 different groups, Janet Rowley's group, Carlo Croce, Mike Cleary, and Stan Korsmeyer, all cloned the 11q23 breakpoint and found the gene that we now most often call MLL. It had a number of different genes there at 11q23. Then in the mid-90s, Stan Korsmeyer's group demonstrated that the wild-type MLL1 gene, in this case, in knockout mice, controlled homeotic or Hox gene expression during axial skeletal development in this set of studies, and ultimately also in hematopoietic development. This was important because there was already a gene called trithorax that had been described in Drosophila that had been shown to control axial skeleton or skeletal development and also control homeotic gene expression. And the reason that it was thought that these 2 genes might be homologs was this C-terminal set domain shown here, which at the time was thought likely to be a histone modifying domain, but had not been shown yet. And then in 1996, Mike Cleary and Terry Rabbitt's groups, using different mechanisms, one a knock-in mechanism and one a retroviral overexpression mechanism, demonstrated that the MLL fusion protein generated by the MLL translocations were indeed oncogenic. And at that point, we learned that it, of course, wasn't only the 411 rearrangement, but there were many MLL rearrangements. We now know that there are over 70 or 80 different rearrangements, all of which occur with the 11q23 or MLL, and generate these fusion proteins that are demonstrated here, where the N-terminus of wild-type MLL is fused to the C-terminus of one of these 70 or 80 different translocation partners. And will point out, however, that a few of the translocations are particularly common. MLL-AF4, which is the product of 411 rearrangement, MLL-AF9, which is the product of 911 rearrangement, MLL-ENL, 1119 rearrangement, and MLL-AF10 and MLL-ELL account for somewhere on the order of 80% of the translocations. So there's something about these fusions that makes them particularly relevant, and we'll talk more about that in a minute. So at that point, we knew that the MLL translocation generated a fusion protein that was oncogenic, and that wild-type MLL controlled homeotic or Hox gene expression. And many of you probably know that Hox genes encode for the homeotic or Hox proteins, which are DNA binding transcription factors, again, important for control of developmental gene expression. Then when I was a fellow in Stan Korsmeyer's lab, about the same time that the St. Jude group led by Jim Downing demonstrated that MLL rearranged leukemias have a very distinct gene expression profile as compared to other subtypes of either ALL or AML. I did this with then the state of the art, working very closely with Todd Golub to do microarray analysis similar to what the St. Jude group had done. And importantly, we were able to show that the Hox genes, in this case, the Hox A cluster genes, which are the genes that are most relevant in hematopoietic development, are expressed at relatively high levels in MLL rearranged leukemias compared to those without MLL rearrangements. And the importance of this was that it suggested that the fusion protein was actually driving gene expression or maintaining inappropriate gene expression in the leukemia, which up until then was a bit of a question because it wasn't so clear whether or not these were gain of function or loss of function mutations. However, at that point, it still wasn't clear what the MLL fusion protein was doing and what MLL itself actually did, even though it was known that it bound to chromatin. but not in a sequence-specific fashion. So it was still a bit of a conundrum until two papers published simultaneously, one by Tom Milne when he was in Jay Hess' lab working with David Alice, and another by Nakamura and Eli Kanani and Carlo Croce's lab, isolated the wild-type MLL1 complex with multiple proteins bound to MLL1. That's important because up until then, enzymatic activity had not been able to be shown. But when the full protein complex was purified and isolated, MLL1 did indeed have histone H3 lysine 4 methyltransferase activity. And that was shown to be through this C-terminal set domain. That's the enzymatic portion of MLL. So indeed, the MLL protein is a chromatin regulator, and therefore MLL fusions very likely influence gene expression via dysregulated chromatin control in one way or another. I haven't pointed out yet that the wild-type MLL, which is shown here, is a quite large protein. It's nuclear, as I suggested, with a number of different domains, and all the function of these various domains is still under a fair amount of investigation. So that led us to the following question that really gets back to some of the questions that I was alluding to earlier, and that is, when the MLL fusion is expressed, when the translocation is generated, what happens to both gene expression and the potential cells of origin when that fusion protein is found in the context of a hematopoietic cell? And that was the question we started addressing in 2006, and over about 10 years worth of work have shown that you can introduce the MLL fusion protein, in this case via retroviral mediated delivery, into either hematopoietic stem cells, as I've already said, the cell type that gives rise to all hematopoietic cells and is able to self-renew, or into more downstream committed progenitors like the granulocyte macrophage progenitors shown here, which does not have long-term self-renewal potential. If you inject these into a recipient mouse, they'll give rise to more differentiated cells, but they will not hang around for more than a couple of weeks. But when you introduce the MLL fusion into these cells and inject them into a mouse, invariably the recipient mice will come down with leukemia, where the apex of the leukemia hierarchy, the leukemia stem cell, is a cell that looks most like a progenitor that's inappropriately expressing gene expression programs normally highly expressed in the normal hematopoietic stem cell. So the point is that during normal development, there is a program of gene expression that's being gradually suppressed or repressed as cells move down, in this case, the myeloid pathway. And the MLL fusion is able to interrupt that, re-express those genes to high level. And the idea is that's what generates the long-term self-renewal potential of the leukemia stem cells. And in fact, some of those critical genes are direct MLL fusion targets. We now know, based on this, our work and many others, that the MLL fusion protein binds to chromatin in and around the HOX-A cluster and to a number of other genes, including a gene called MEIS1, which encodes for another DNA binding protein that heterodimerizes with the HOX-A cluster. We've also found that the beta-catenin pathway is activated in a number of other canonical stem cell type pathways. Again, the concept being now you have both self-renewal potential and stem cell associated programs expressed in a cell type where they're not normally expressed, and that is driven by the MLL fusion and leads to constant self-renewal and ultimately leukemia development. With this type of data in hand, we've been able to compare that to a number of different mouse models and to different human leukemias. And I would say we have a very, very good understanding, probably as a result of this model, maybe the best understanding of any type of leukemia, perhaps any type of cancer, where a DNA binding protein binds to specific sites on chromatin and controls a very specific gene expression program. That allows us to start to ask fairly sophisticated questions. I point out here a few transcription factors that we know are both interesting and essential in the continued proliferation of these leukemias that's come out of this work and others. So that leads to the question as to, okay, that's what happens. How is it, in fact, that the MLL fusion disrupts normal transcriptional control to either drive or maintain gene expression and leukemia development? So that leads us to what we know about chromatin-based regulation of developmental gene expression. And what I mean by that, in this particular case, are the Hox genes and MEIS1. Again, genes that are normally expressed in stem cells and being shut off during myeloid development. And 3 different chromatin-associated complexes are shown here. Of course, there are many others, but these are 3 that are relevant for today's discussion. The MLL complex here on the left, there's MLL1 itself, another protein component of the complex menin. I'll talk more about that later. WDR5, a number of other associated proteins. And this complex places the histone H3 lysine 4 trimethyl modification associated with many, if not most, actively transcribed genes. A second complex, the superelongation complex shown here, the enzymatic component of this complex is CDK9, which phosphorylates RNA polymerase II on its C-terminal tail to allow for transcriptional elongation, and the DOT1 complex, which places a different histone modification, H3K79, either di or trimethylation. This modification is also associated with most highly expressed genes. The reason I point these 3 complexes out is 1, because MLL, of course, is where the breakpoint is, and 2, many of these proteins in these 2 complexes are found as MLL fusion partners in the translocation, or in leukemia translocations. ELL, AF9, AF4, AF10, and AF9 are all translocation partners with MLL, suggesting that you, in fact, fuse, if you will, the MLL complex to a couple of different other chromatin-associated complexes that normally control gene expression. And it's probably this dysregulation that leads to aberrant gene expression. So there's a lot of work been done to show all of that. I'm obviously not going into the details, but it does point to potential therapeutic opportunities in the proteins of these various aberrant complexes. So I've just listed a few here. The first 2 are enzymes. The reason for that, obviously, is small molecule inhibitors of enzymes is a tried and true path to drug development. And DOT1 I'll talk briefly about in a minute. That's the histone H3 lysine 79 methyltransferase. CDK9 is the kinase that phosphorylates RNA polymerase II. Molecules have been developed to target CDK9. They tend to influence gene expression broadly, and still being worked on to understand exactly how one might use those. And then there is the potential, and I'll talk more about the MLL-menin interaction in a minute, to disrupt some of these protein-protein interactions, WDR5, BRD4 inhibitors, as many of you have heard about. And again, I'll mention some of those later. So the first gene that we started studying in these complexes that might have a potential role in MLL fusion-driven leukemias is DOT1. And I mentioned DOT1, I've talked about it in other settings, but we did learn a fair amount from the study of DOT1 and ultimately the development of small molecule DOT1L inhibitors. And Catherine Berndt, when she was a postdoctoral fellow in the lab, generated a conditional DOT1L knockout mouse, transformed the bone marrow from those mice with MLL-AF9. The controls are shown here. This is a leukemic colony. These are leukemia cells. And when you inactivate DOT1, the colonies really don't replate. Cells become distributed throughout the dish and look very differentiated, and these cells will not induce leukemia. Importantly, in one of the first pieces of data that suggests there might be some selectivity of the importance of these proteins or these chromatin-associated proteins to MLL fusion leukemias, is that if we took HOXA9 and MEIS1, both of which are known to transform mouse cells on their own, express them in the bone marrow of these DOT1 conditional mice, and then inactivate DOT1, leukemia cells proliferate normally. That is, DOT1 is not required for proliferation of all hematopoietic cells. And that was critical as we started to think about the therapeutic opportunities of this and other approaches. The other piece of information that was important was that when we inactivated DOT1, the genes that were most sensitive to that, shown, some of which are shown here boxed in red, were indeed the MLL target genes, the genes being driven by the MLL fusion. So DOT1 was not required for all hematopoietic proliferation, and the genes that the MLL fusion is driving are particularly sensitive to DOT1 inactivation. So we've done a number of studies looking into how this may work, and it appears that what DOT1 is doing in this context is inhibiting repressive mechanisms, some of which are shown here, a deacetylase and a histone methyltransferase, that would normally shut off these programs at this stage of hematopoietic development. And if you inactivate DOT1, then the gene expression decreases. An important point of that statement is the gene expression decreases. Not in all cases does it completely shut off, and that's probably relevant as we move forward. So this, while we were doing this, we were working with Epizyme, then a relatively young company, to develop small molecule inhibitors of the DOT1 methyltransferase, and the structure of DOT1, or at least the enzymatic portion of DOT1, bound to the clinical candidate EPZ5676 is shown here. And that has now gone through phase 1 clinical trials a number of years ago. And in the phase 1, there was no maximally tolerated dose reached, so the molecule was not particularly toxic at all up to the doses that were given. About 25% of patients showed an increase in their absolute neutrophil count, not just a mild increase, into the 20,000 range, which is extraordinarily high. And we sorted those neutrophils from a number of patients and demonstrated that the MLL translocation is indeed there, suggesting we were getting a significant amount of differentiation of the leukemia cells in patients, which is what we would have expected based on the preclinical and the knockout data. And 2 of the 30 or so patients in the trial did go into complete remission, one with quite a long CR, this patient here who also had leukemia cutis, but did have involvement of her bone marrow as well. So biological activity, but as a single agent, not enough activity to move forward. And so therefore there's the thought about combinations. What we learned from this is that responses take time. Can take this patient in particular, took over a month in order for her to go into remission. In these samples where we were able to assess it, DOT1 was inhibited to about 60 or 70%. That may not be enough. We now know that we need deep and constant inhibition. That could have been a problem as well. I mentioned combinations and minimal toxicity. One thing we've learned since, and this is important as we talk about the MLL-menin inhibitor, is that with the enzymatic inhibitor in vitro, so these are MLAF9 transformed cells growing in vitro, these are the control treated, and these are the DOT1 inhibitor treated cells. The cells arrest, they differentiate, some die, and then over time they'll start growing again, back up to the rate at which the control cells will grow, and we know the enzyme is still inhibited. So it is the case that the cells can become independent of the enzymatic activity of DOT1. So that's important to think about as we think about combinations, potential mechanisms of resistance, and so on. And I think what we've learned over time, and again, I'll show you some more of this in a minute, is inhibiting any single enzymatic activity in a large multi-protein complex may not be enough in order to get the types of responses that we're interested in. So along those lines, we started thinking about other possibilities in the complex. Needless to say, there's much work going on looking at the various proteins and protein-protein interactions. And that led us to focus next on the interaction between MLL1 and this protein menin. So the amino terminus of MLL fusions binds to a pocket on the MEN1 protein. So MEN1 protein is encoded by the multiple endocrine neoplasia type 1 gene. So this is a gene that's a tumor suppressor in other settings. This is the crystal structure of MEN1 with a small molecule, one that I'm going to show you more details about, shown here, VTP50469, bound to a pocket where the N-terminus of MLL, shown here, is usually bound. So these 5 amino acids here, FPARP, of MLL bind deeply into this pocket of menin, and it's been shown that that interaction is critical for continued MLL fusion-driven leukemia growth and proliferation. So we started working about 5 years ago with a company then called Vitae, Jerry McGeehan at Vitae, now at Syndax, to develop potent and selective small molecule MLL menin inhibitors. And here's the concept that was originally proposed by Mike Cleary based on his work, Jay Hess's work, and work from Matthew Meyerson originally, demonstrating that menin bound to MLL complexes. And the concept is these molecules would disrupt this interaction. The MLL fusion then would be unable to continue to drive a homeotic or Hox gene expression. So with this molecule, we were able to show that it's quite potent in inhibiting proliferative activity of a number of MLL rearranged lines. You can see in the low nanomolar range. And if you assess this molecule on any other cell line other than those with NPM1 mutations, and I'll tell you about that in a minute, they don't have much in the way of anti-proliferative activity. Again, this is the structure of the molecule. And if you look at menin on size fractionation columns or glycerol gradients as shown here, it usually runs in multiple fractions, including in the high molecular weight fraction. When you treat with the inhibitor, now all of the menin moves out of the high molecular weight fraction and into the free protein fraction, showing that indeed you've disrupted the menin interaction with what are presumably transcriptional complexes. And the gene expression changes shown here at day 2 or day 7 in an MLL rearranged AML line. You see a loss of expression of genes that we know are directly regulated by the fusion, which are shown here in red at day 2 and day 7. So menin's coming out of complexes, and we're getting relevant changes in gene expression, and those lead to selective inhibition of proliferation in leukemia lines. The leukemia line proliferation data would be very similar to what you would see with a DOT1 inhibitor. Here's what's different between the menin inhibitor and the DOT1 inhibitor. So this is ChIP-seq, shown both by a tornado plot and looking at selected genes. This is ChIP-seq for menin. So I've shown you that menin's out of the high molecular weight complexes. This now shows you that menin has largely come off of chromatin here when you treat with the inhibitor. Interestingly, and a surprise from this is that the MLL itself globally is not lost from chromatin. So you can see here, and in fact, this was a surprise because many of the models suggested that disruption of the MLL-menin interaction should affect MLL broadly. That might be toxic if you did that, but in fact, that doesn't appear to be the case. You see a decrease in DOT1 occupancy. It's probably due to destabilization of the DOT1 protein, and at least immediately you don't see global changes in K79 methylation, which is regulated by DOT1. The most interesting part comes when you look at specific loci. So here's the HOXA cluster, and we'll focus on MEIS1 here. So menin is gone from chromatin when you treat with the inhibitor. That's to some extent expected. But interestingly, MLL, in this case, the MLL fusion, is lost from MEIS1 here, but not lost from the MLL locus. And that actually goes along to some degree. And the take-home point here is that you treat with the menin inhibitor, menin comes off of chromatin globally, but you get selective loss of MLL, and there's very tight association between the genes that lose MLL and decreases in gene expression. So the interesting finding, and perhaps important maybe for therapeutic window, is that you're really affecting a very selected group of loci when you treat with the inhibitor. So with this inhibitor, which is quite stable in mice, has very good PK properties, we were able to do a number of preclinical studies, which are highlighted here. The small molecule can be put into the mouse chow. We injected mice with PDX models of MLL rearranged leukemia or the mouse model and studied survival. This is just a survival curve of an MLAF9-driven mouse AML. The mice were treated, as you can see here, for about 30 days, and you see a dramatic prolongation, in fact, probably eradication of the leukemia in the mice. So the remarkable aspect of this is a very aggressive mouse model of AML. Many of you probably that work on leukemia have probably worked with it. And this is a pretty dramatic response in terms of increase in survival. So we now moved on to work with Richard Locke in Australia, who had a number of MLL rearranged ALL PDX models, which he was able to do these preclinical studies in that setting. And he saw a significant survival benefit in 7 of the 8 PDXs after a single 28-day treatment with 469, shown here. Profound effects on MLL1 and MLL2, 2 different models. You can see some of those mice, really the disease was eradicated. And this was a BCR-ABL rearranged ALL where there was no effect. And there was only 1 MLL rearranged ALL, number 5 here, that didn't seem to respond. So that's the data with the MLL fusion. That is that either DOT1 inhibitors or menin inhibitors have pretty dramatic in vitro effects. The menin inhibitor really seems to have more dramatic effects in PDX experiments. And what I didn't say is that while the gene expression changes ultimately are very similar, the menin inhibitor really seems to lead to much more rapid changes in gene expression than the DOT1 inhibitor, and we're starting to think about the mechanisms there. So I'm now going to shift gears to, as we started to think about how might we use some of this information we're finding in the setting of MLL to think about and potentially understand and treat other subtypes of AML. So I've already told you that the MLL, a central part of the MLL fusion gene expression program is high-level expression of Hox genes and this protein MEIS1. This is a gene expression paper and genomics paper from Tim Lay's group characterizing a number of subtypes of AML. And here you can see high HOXA level expression in the MLL rearranged leukemias, high MEIS1. There's another large subgroup of AML here where you see high HOXA, HOXB, another group of HOX genes, and MEIS1 expression. And about half of those also have mutation of the nucleophosmin-1 or NPM1 gene. And so this led us to start to think about whether or not these complexes might be relevant in the maintenance of the gene expression of this subtype of AML as well. This subtype is much more common than MLL rearranged leukemias. It's about 30 to 40% of adult AMLs broadly, so many more patients with this subtype. The mutant NPM1 protein is cytoplasmic. The mutation leads to movement, if you will, of the nucleophosmin protein out of the nucleus into the cytoplasm. NPM1 is usually in the nucleus, nucleolar. Its specific function, not so clear, a number of functions ascribed to it, but clearly the mutation leads to a cytoplasmic NPM1 protein, and it tends to occur with other mutations like DNMT3A. So there are a number of groups working on the mechanisms of NPM1-driven AML. The molecular mechanism is still not particularly clear, but Peggy Goodell's group recently did a very interesting set of experiments where they used CRISPR to re-engineer, if you will, the mutant NPM1 so that it moves back into the cytoplasm. That led to differentiation of those cells and a decrease in HOX, MEIS, and a number of other genes, suggesting that there is, in fact, a relationship between the NPM1 mutation and HOX, MEIS gene expression. And that is in line with work that we had published a couple of years earlier, Michael Kuhn's work, where he had demonstrated that the MLL1 gene, or the MLL1 protein, is important for the proliferation of NPM1 mutant AML, both human cell lines and mouse models. He initially identified this using a CRISPR domain screening approach, originally defined, described by Chris Vakuch's group, where you design specific CRISPR guides to target regions of the gene that encode for specific domains of the protein. And he found that the region of MLL that binds to menin, shown here, it was particularly sensitive to CRISPR-mediated mutation. And he went on to show with some probe compounds developed by Jolanta Grimbecka's group, and I should point out she's been really working in this field for quite some time, that indeed those probe compounds had some anti-proliferative activity. And the concept here is much like in the setting of the MLL fusion, the molecule disrupts this interaction between MLL and menin, perhaps the disruption of the interaction of wild-type MLL with menin in the setting of NPM1 could lead to important changes in gene expression, ultimately differentiation and decreased cell growth. While we were doing this, a couple of papers were published by John Dick and Ravi Majeti's group, starting to highlight some of the other aspects of NPM1 and DNMT3A mutation biology. And what they were showed in these two papers is that DNMT3A mutations almost invariably in adults with AML are found in the hematopoietic stem cell compartment, whereas NPM1 mutations are most often found in the progenitor compartment in fully developed AML. And the suggestion perhaps being that, and knowing that based on data I'm going to talk about in a minute, that the DNMT3A mutation comes first, and perhaps the NPM1 mutation occurs in a downstream myeloid progenitor. So if we think about that in the context of the evolving understanding of clonal hematopoiesis that you've already heard about from, if you've been listening to these, from Ross Levine and Ben Ebert, where as people age, they tend to develop clonal hematopoiesis with clones that are derived from hematopoietic stem cells that have mutations in genes that are frequently found in AML, like DNMT3A, TET2, ASXL1. Those individuals actually do not have often obvious changes in their hematopoietic parameters, but over time they may acquire other mutations like NPM1, RUNX1, CBP alpha, and then ultimately progress to AML. Importantly, these mutations are not found in clonal hematopoiesis, neither are FLT3 and signaling RAS mutations, suggesting that perhaps these mutations are doing something different and perhaps they're occurring in a different cell type. So with that question in mind, Hannah Ockelman got mice from Ross Levine's lab that have a conditional DNMT3A mutation and a conditional NPM1 mutation, and crossed them to ask the question as to whether or not either NPM1 or DNMT3A or the combination could induce stem cell-like properties in myeloid progenitor cells, much like we had found in the MLL setting. And if so, what are the genes, proteins, and complexes important in that setting as well? So she generated these strains of mice, wild-type, single mutant or double mutant mice, and then either injected PIPC to activate these mutations or used a retroviral CRE in specific cell types and then looked at gene expression. So this is the PIPC experiment from mice with the genotypes shown here. In the black and the gray, you can see HOXA9 expression is decreasing throughout myeloid development, as I've told you before that is the case. Whereas if you have an NPM1 mutation or the double mutation, HOXA9 expression is maintained at a high level throughout hematopoietic development or throughout myeloid development, and you don't get repression of HOXA9 as you normally would. If you do RNA-seq on this, in fact, much of the HOXA cluster and some of the HOXB cluster shows the same pattern. So you have inappropriate expression of the HOXA cluster in the granulocyte macrophage progenitors. And if you look at a more broad hematopoietic stem cell signature, it's inappropriately expressed in the progenitors, much like we had seen in the setting of the MLL fusion. I'll point out that those gene expression changes look very similar if you use an approach where you isolate the granulocyte macrophage progenitors first and then activate the mutation, showing that the mutation can impose upon the progenitor a gene expression program like I just showed you, rather than having to maintain it from the hematopoietic stem cells. So what about the function? So I'll just show you, she again did this by injecting PIPC or using the retroviral CRE method. I'm just showing you the retroviral CRE here. If you take wild type GMPs, in this case, they will not replate in vitro. If you take DNMT3 mutant GMPs, they also do not replate in vitro, whereas the NPM1C and the double mutant will replate for 3 or 4 weeks in vitro, suggesting there is some degree of long-term proliferative capacity in these progenitors that are either expressing the NPM1C alone, or the combination. And then if you inject those cells into mice, in fact, the NPM1C or combination will lead to enhanced engraftment and long-term proliferation of those GMPs in mice. So now the question is, do those generate leukemia? So up until this point, what I've said is NPM1 mutation in granulocyte macrophage progenitors or DNMT3A and NPM1 mutation makes the progenitors proliferate in vitro long enough that we can see them for many weeks, up to 16 weeks at least. Are they serially transplantable? Do they have true self-renewal potential? So we took all of those primary mice, injected the sorted granulocyte macrophage progenitors into secondary mice, and all of those mice ultimately came down with leukemia. So the point here is that the progenitors are self-renewing for a long period of time before ultimately in a secondary recipient they will develop AML. And this shows that the GMPs can indeed act as a cell of origin, and there's a significant window of time between when those GMPs are self-renewing and ultimately AML develops. So here's our current thought on how this works, and that is that the DNMT3A mutation occurs in hematopoietic stem cells. That's pretty clear. You get clonal hematopoiesis and multilineage differentiation of those DNMT3 mutant hematopoietic stem cells, and ultimately the NPM1 mutation is acquired in the DNMT3A mutant, in this case progenitor. That leads to a self-renewing leukemia stem cell with inappropriate expression of the HOX and MEIS genes in a myeloid progenitor, much like we saw in the setting of the MLL fusion. So the question is can we target this gene expression as well? As you might imagine, where we're headed is to look at the MLL menin inhibitor that I've already shown you in the setting of the MLL fusion to see if indeed it has similar activity in the NPM1 mutant setting. So this is the only easily propagated NPM1C mutant human AML cell line, OCI AML3. And you can see with the increase in concentration, you get dramatic inhibition or proliferation of this cell line. You see a similar effect on mouse NPM1 mutant cells growing in vitro. So this is colony replating. You see that's inhibited over time with the MLL menin inhibitor. This is the same inhibitor I described that we used in the setting of the MLL fusion, and you get some degree of differentiation. So indeed, this inhibitor does have anti-proliferative activity on NPM1 mutant AML lines. If we look at gene expression, that's shown here. The most dramatic changes are in genes such as MEIS1. I've told you already that's the same in the MLL fusion, and another DNA binding protein called PBX3 that also binds to the HOX protein. So those are quite dramatically changed. The HOX genes themselves changed modestly, but not consistently and not dramatically. So there's a little less effect on the HOX genes in this setting, but quite a significant effect on these other DNA binding proteins. So we focused on MEIS1, knowing as a result of CRISPR screens and other types of approaches that MEIS1 is likely to be an important gene in this subtype of AML. So we did a CRISPR of our own, and indeed MEIS1 inactivation decreased colony formation of the NPM1 mutant AML cells. And importantly, if we can rescue the proliferation inhibition by the menin inhibitor just by re-expressing MEIS1 off of a viral promoter that's insensitive to the menin inhibitor. So indeed, MEIS1 expression does rescue the anti-proliferative effect of the inhibitor. Another point to make that reminds me to make a broader point is that when you treat with the menin inhibitor, here's labeled VTP, you see a significant decrease in the menin protein level. And this is not as a result of change in RNA. And that can be reversed at least to some extent by treatment with the proteasome inhibitor. And we've seen this, and as have a number of others. And what we think is happening is the menin inhibitor is disrupting to some extent the whole protein complex and destabilizing at least menin and probably other components of the complex. And I say that because that is probably part of the reason that the menin inhibitor is having more rapid effects on gene expression as compared to the DOT1 inhibitor, is we're really having a more dramatic impact on the chromatin-associated complexes with this small molecule. So if you look at chromatin changes in the setting of an NPM1 mutant leukemia, really summarize all of this to say it looks very much like what you see in the setting of the MLL fusion. That is, you lose menin, You don't lose MLL occupancy globally. However, you do lose MLL occupancy at certain loci, such as MEIS1 shown here. And those loci are the ones where you see dramatic changes in gene expression. And here's the GSEA of that correlation. That is, menin's lost everywhere, MLL's lost at certain genes, and those genes are the ones that are sensitive in terms of their gene expression. So again, some selectivity in terms of locus response to the MLL-menin inhibitor. This is just a set of studies showing that indeed the menin knockout resembles the inhibitor. That is, the guide to MLL1 or menin both suppress MEIS1. Guide to MLL1 suppresses Hox gene expression, but a guide to menin does not suppress Hox gene expression, and I've shown you that's what happens with the inhibitor as well. So what if we move in vivo? I already told you this small molecule has very nice pharmacokinetic properties. I've also told you there's a period of time when these progenitor cells are self-renewing in the mice before they ultimately develop AML. So that led to the experiment where Hannah wanted to see if she could intervene at that time when the progenitors were self-renewing, but before the mice developed leukemia. So she injected a number of mice with what we call long-term GMPs, made sure they had engrafted, and then watched engraftment over time in the control, and then ultimately in these secondary recipients, those mice will die of AML. Whereas if you put them on the small molecule MLL-menin inhibitor, you see a dramatic loss of engraftment, and ultimately those mice will not develop AML. So she's able to intervene prior to the development of AML and prevent the progression to full-blown AML with this small molecule. And you can see here the mice were on the molecule for quite a long period of time and remarkably had very few, if any, hematopoietic side effects from the molecule. So this can prevent development of AML from these progenitors. So this is PDX data from human AMLs, two different human AMLs, one with an NPM1C mutation and a FLT3 ITD, another with NPM1C, FLT3 ITD, DNMT3A, and IDH. This one, we started treatment after we noticed 1% engraftment. That's the standard way to do the experiment. And you can see we were actually able to eradicate the disease. Remarkably, and this is a rare molecule that can do this, in one set of experiments, we let the mice really develop fulminant disease to the point where they were going to die in a few days, and then started them on the treatment. In fact, we were able to rescue 5 out of the 6 mice in the treatment arm on the small molecule. So pretty dramatic PDX activity. It leads to the question that we don't know the answer to yet, but I thought it would be interesting to show is, so why is it that are the leukemias so sensitive to this small molecule, but it doesn't seem, at least in initial studies, that normal cells are particularly sensitive, because the MLL-menin interaction probably controls, we thought, Hox and MEIS expression in wild-type stem and progenitor cells as well. So this is NPM1 mutant GMPs treated with the inhibitor. I've already shown you MEIS1 changes dramatically. Some other interesting genes, EVI1, IKZF2. Interestingly, though, in the wild-type cells, if you look at MEIS1, they really are not. It's really not particularly sensitive to the inhibitor in the wild-type. That's LSK stem and progenitor-enriched cells. Not much of a change. There are some other changes, but the point is that the leukemia cells seem to be way more sensitive in terms of gene expression changes to the inhibitor than wild-type cells. The molecular mechanisms for this are as yet not understood, but certainly will be interesting to dig into. So to kind of summarize what I've been talking about here, wild-type HSCs develop mutations that lead to clonal hematopoiesis. That's been studied and shown. Very interesting work from Ben Ebert, Ross Levine, Ravi Majeti, a number of others. Over time, you acquire other mutations, we think, in the progenitors, and those mutations are the ones that give you the fulminant AML. And there may be, we don't know yet, it's so well in humans, the studies are just being done, a period of time here where you potentially could intervene if you had a relatively non-toxic treatment to prevent the development of leukemia. And needless to say, more work to be done there, but a developing concept. Ultimately, if you had molecules to inhibit clonal hematopoiesis, you could do that as well. However, these patients are not guaranteed to develop leukemia, so the bar for such a therapy would presumably be higher. So I'm going to end with just a couple of slides, a few slides on the initial data from the clinical trial of what's called Syndex 5613. This is a similar molecule to 469, orally bioavailable, and in the clinical studies treated BID. So this is just showing Kaplan-Meier survival curves for this molecule in a MOLM13 disseminated model. I won't dwell on this too much other than to say that there is a dose response, and most of the studies that I've shown you for 469 are likely to be repeated for this molecule. And you see a decrease in human leukemia as assessed by human CD45. So just a lot of work went into looking at dose response and levels of the drug required to get those responses. I won't go into that detail here. The clinical trial has started. It's a phase 1 dose escalation trial enrolling patients with relapsed refractory acute leukemias. This is the dose escalation plan, and then ultimately there would be a phase 2 expansion with the specific subtypes of AML. I think 6 adult patients have been treated and 5 pediatric patients have been treated with them or enrolled into the trial. It's still very early, just opened a couple of months ago. And I'm pointing out here the ones where we know what dose levels have been attained in those patients, because via modeling, Syndex has been able to predict what level should be required in order to see a response. The first, as you might imagine, couple of patients here did not meet the PK profile target, which is not surprising given that you started a relatively low dose. 5 pediatric patients have been treated as well. They also did not get to the plasma concentration expected for a response, and none of these patients responded. However, one patient, second patient enrolled on this dose, is refractory mixed lineage leukemia with an MLL TET1 translocation and a FLT3 ITD, had been through chemotherapy and gilteritinib treatment. Again, this was the dose of the small molecule. There was no DLT, but there was a grade 2 QTc prolongation, and so therefore the dose was decreased. However, this patient was also on an antifungal, which inhibits cytochrome P450 enzymes, and it was known that this molecule was metabolized by those enzymes, and this patient had a much higher level of plasma concentration of the molecule than would have been expected at this dose, and above the levels predicted to be required for response. And in fact, this patient went into a CRI, a complete remission with incomplete hematologic recovery, and then when the dose was back down, went into a CR and continues on the drug for now. So the point being, the one patient that's gotten to the plasma levels predicted to be necessary for response, in fact, went into a CR. So it's very early, but the trial is ongoing, and there are some possible interesting results there. So to summarize this part, this is a potent selective orally available inhibitor, Syndex 5613. I didn't go through the biopharmaceutical properties. It does appear to have monotherapy activity in multiple preclinical models. We're still working on some of that. Pharmacokinetics appear to be affected by CYP3A4 inhibition that led to relatively high levels in one patient and a clinical response in that patient, and the clinical investigation is ongoing. So now to summarize the broader points, I've shown you that NPM1C and MLAF9 induces self-renewal properties in myeloid progenitor cells during progression to AML, and that menin removes MLL from specific loci that are critical for continued proliferation of the leukemia to reverse gene expression in those progenitor cells, leading ultimately to differentiation. An interesting point for the field is that the HOXA cluster gene expression is only modestly affected by menin inhibition and only slightly more by DOT1 inhibition, and there really seems to be some locus-specific responses, both in terms of gene expression and chromatin-based responses, and what the mechanisms are for those is still under investigation. and pre-leukemic and leukemic NPM1 and MLL rearranged leukemias can be eradicated by menin inhibition in vitro. So I'll stop there. Just point out before I do that many of these concepts are now being looked at in various types of sarcoma by ourselves and others, and hopefully we'll see similar types of responses there. That's yet to be seen. Those molecules are still a little bit further back. And then, of course, more broadly, in various types of adult solid tumors that are driven by aberrant gene expression, one can imagine thinking about these approaches as well. So I didn't mention most of the people involved as I went along. Have a very motivated and committed group of people in the lab who have done much of this work, and really enjoy working with them and now Zooming with them over the past couple of weeks. Here are some of our collaborators and our funders, and I think I'll stop there. Thank you.
Well, Scott, that was just perfect. Thank you. Very inspiring stuff, and the clinical trial is definitely exciting. There are a number of questions. We won't have time to get to too many of them, but I'll start with a couple that bring together questions that were posed by the audience. The first is, are you surprised that MEIS1 overexpression is sufficient to overcome the effects of the menin inhibitor? That it's just as simple as everything goes through MEIS1?
Yeah. It's. I don't know if I'm surprised by the result of the experiment, but I do agree that it probably doesn't tell us that it's as simple as MEIS1. So we know MEIS1 is important in this leukemia just from genetic inactivation studies. And so in that sense, I think it's the result of the experiment is not surprising. Having said that, with these, we can show similar types of results with HOXA9 and MEIS1 other settings. And I do worry that relatively high level expression of those transcription factors may overcome many different things that are being affected by the inhibitor. So I think what I'm saying is that MEIS1 is important. I think that does tell us that that's an important part of the equation. I do think menin is likely to be doing other things and working with other proteins as well, but that's in that setting, MEIS1 is enough.
So you mentioned some of the shortcomings of enzymatic inhibition and the failure of the complex to fall off the chromatin at all the sites. What about degraders? If you degrade some of these same factors that you can enzymatically inhibit, does the complex fall off the chromatin?
Yeah, so that's a good question. We're just starting to do those types of, we don't have small molecule degraders for many of these. But if you genetically inactivate DOT1, you do see more dramatic changes at chromatin. We haven't yet for the MLL menin inhibitor seen, if you genetically inactivate menin, which would presumably be the equivalent of degrading it, versus inhibiting the MLL menin interaction, we haven't seen differences yet there. And I don't know if that's just because disruption of a protein-protein interaction may be more dramatic and look therefore more like a degrader. But the general concept that if your small molecule has only modest effects when you inhibit either an enzymatic activity or a domain, will turning it into a degrader lead to more dramatic effects. We have seen that in other cases like BRD9, where the inhibitors, the bromodomain inhibitors, don't do much in the setting of synovial sarcoma, but the degraders have dramatic activity. So I do think as a rule, that may be a possibility.
Another question, the MLL fusions are such potent oncogenes, they may be able to transform as a single hit. Any insights about what it is about a GMP that makes it so permissive and why MLL fusions aren't seen across other cancer types?
Yeah, that's a terrific question. So the short answer is, I don't know. The longer answer is, we actually have shown working with Dan Tinnen that it really is something about the GMP that's particularly relevant. That is, if we prevent, the relatively hard experiment to do, but if we prevent differentiation to the GMP stage, you actually prevent leukemia development. So there is what we think is a true interaction between the chromatin state in the granulocyte macrophage progenitor and the MLL fusion protein. But what that specific state is, we don't know.
Well, since you mentioned Dan, we'll give him the last question. His is, I'll read it. Do you
Oh,
think the
great.
length of time for response to DOT1 inhibitors and DNA methyltransferase 1 inhibitors reflects epigenetic modifiers as enforcers rather than drivers of transcription?
I think I agree with the concept that epigenetic regulators may be enforcers. Yes. Does this result support that? I guess so. But I do agree with that concept. I think they're often, if not most of the time, maintaining relevant gene expression. That doesn't make them any less relevant therapeutic targets, but I assume what he's alluding to is that the transcription factors probably are the drivers, and I think I would agree with that.
Well, thank you again for just masterful talk. Again, we'll not have a lecture tomorrow, but we'll reconvene Thursday at noon Eastern Standard Time for Dr. Marcus Munchin. And until then, everyone, please be well.